on the behalf of Learn to Create, to welcome you all for the today's session webinar, which is on cancer biology. Okay. So the session has been divided into just two parts. The first part, I will be just giving you the fundamentals of what is a cancer biology and why it is becoming one of the important course because there are a lot of cases of this chronic disease is increasing. So that's why there are a lot of research is going on in this area, that is cancer biology. Okay. And this second part will be dealing with the scope and opportunity if you want to excel in this sector or in this area or in this domain of cancer biology. So I will giving you how you can proceed with different job opportunities, or if you want to work in the research lab, how you will be applying and how you will be getting the jobs, or what is the job scenario. So good evening, everyone. I'm getting the messages. Okay, so let us start the session. Okay. So you all have come across this term chronic disease. Have you heard about this term chronic disease? Do you know what is chronic disease or disease of chronic diseases? Have you heard about this term? Yes or no? Just reply in the chat box. Have you heard about this term chronic diseases? Okay. So just I'm asking, is COVID a chronic disease? Is COVID a chronic disease? COVID is the chronic disease? Yes or no? No, right. So see, when we are talking about the chronic disease is a condition, okay? It's a condition where the disease or the symptoms which you are facing last for more than 1 year, okay? So whenever a disease or condition that usually lasts for 1 year, or you can say for 3 to sometimes few definition includes 3 months or 5 months also, but disease or conditions that usually last for 1 year or longer, then we call it as a chronic disease. And with time, like with with the age, this chronic diseases and its symptoms get worse. Okay, so the disease that is persisting for a long time, then only we are going to call it as a chronic disease. Okay, right, Sakshi, as you have mentioned, the disease which is persisting for a longer period of time, then we call it the chronic diseases. Okay, and normally, sometimes most of the chronic diseases, we cannot cure them. Okay, we cannot cure them. Are you getting my point? We cannot fully cure the person which is facing this chronic diseases. Chronic diseases can only be controlled. Means you are taking a medication, then you can control your chronic diseases. Okay. Similar to chronic diseases, there is a one terminology more that is called NCD. NCD, that is called non-communicable diseases. Okay, so the chronic diseases can also be termed as NCD, that is a non-communicable diseases. Okay, so when I'm talking about non-communicable diseases, you can understand that these non-communicable diseases are a group of conditions that are not mainly caused by an acute infection. So it is not that it is transmissible. Means it can be, one person is getting, other will be getting infected by that. So it is not communicable. It is non-communicable disease. And normally, if we look at the present scenario, you can say it is forming a global epidemic. Okay, global epidemic means every person, because of their lifestyle or diet, they are facing some of this chronic diseases or a non-communicable disease. Okay, so if I just have given you the definition of chronic diseases, can you name some of the chronic diseases or non-communicable diseases which you have heard about? Okay, any communicable or any non-communicable disease, right? So, if you look at this picture which I have given, there are list of chronic diseases. If I include it, there will be a number of chronic diseases. Like we have, most common is a heart disease. If you look at most of the persons, right, most of the persons are facing this problem of heart diseases, and these are hypertension, high BP, coronary artery diseases. So these are all different types of diseases which are included into the chronic diseases. Okay. After that, the other includes that most of you have mentioned, like arthritis. Then you have your diabetes. Diabetes is also one of the chronic diseases which is, you can say, covering a large number of population. Then we have obesity. Then we have oral health problem. Then chronic disease problem. These are all different types of chronic diseases which are nowadays very common in the population. Among these, if we talk about the second largest chronic disease, that is the cancer. Okay, so the cancer is the second leading cause of the mortality after cardiovascular disease. Okay, and if we look at 70% of the world population is facing 70% of the death which is occurring in as per the WHO report, these are because of this non-communicable diseases. Okay, so imagine the level of this disease in the population. So if we talk about the cancer in men, we all know the most common cancers we have is prostate, lungs, bronchus, colon, and rectal. In women, we face most common type of cancer which is present in the women is your breast, lungs, then rectum, uterine, and thyroid. Now you know the most common type of women cancer faces are breast and the uterine cancer, and another major which is becoming the major is our thyroid. Okay. Now, this was just a fundamental about the chronic diseases. Now, the cancer biology, if we look at, it works on only, you can say, one question. It works on one question. What is normal and what is not normal? Because if you look at the research which is going on in the cancer, you will find that cancer is an abnormal cell division. Everyone says it is an abnormal cell division. So how can we define this abnormality? Okay, because cell division is a normal, you can say, metabolic activity which goes in required for the growth and development. Okay, so cell division is a normal metabolic activity which is required for the cell growth and development. But when it takes a you can say a turn where there is an abnormal growth. Okay, so abnormal growth, when it takes place, then it takes the form of a cancer. So cancer is just a disease which occurs at the tissue level, and this cancer is different from normal cell growth in terms of that it is going to divide continuously. Okay, so what happens when a normal cell division works? What happens? The cell will divide, they will go through a normal cycle, and after that they will undergo apoptosis if the damage or repair is happening. Okay, but when the cell takes a form of a cancer cell, means how this division, how this formation from normal cell to cancer cell, we will discuss later. But just to make you understand that when a single cell continuously divides, okay, continuously divides means there is no regulation, means it is not going to stop. Okay, so it is unrestrained manner, means it is a unregulated manner. The cell is continuously dividing. And another important aspect related with this is that when the cell is continuously dividing, it is going to invade, means it is going to invade the surrounding tissue. So when it is invading the surrounding tissue, then what will happen? It is going to, you can say, entering into the metastatic state. Okay, so when it is invading the surrounding tissue, slowly it will be taking a form of a metastatic stage. So when single cell undergoes an uncontrolled division in an unrestrained manner, in an unregulated manner, without going apoptosis, means apoptosis is blocked, means if the cell undergoes a damage and the apoptosis has been stopped and cell is entering into a continuous dividing phase with an unregulated manner and invading the surrounding tissue, then it is taking a form of a cancer. Okay, is it clear? So this is whole concept of cancer biology, where we are going to discuss the study of cancer progression, initiation, and how we can cure this cancer. There are a lot of research is going on in this area just to understand how these cells are taking a form of a cancerous cells, okay, and how this progression is taking place, okay. So if I talk about this cancer, the cancer, they do not look or act like the normal cells, we all know, and this development of cancer is a multi-step process. Okay, it's not a single step that cell is dividing and it will be leading to the cancer. No, it is a multi-step process. The first step in the case of cancer is initiation. Means there will be a cell which will be converted into cancerous cell because of the mutation, and then that is called initiation. So once the initiation has taken place, yes, someone's mic is on. If you have any question and query, you can write in the chat box. I will be responding to it at the end, the questions. Okay. So I was telling about that this cancer cells, they do not act like a normal cell. So first, how this normal cells convert into cancerous cells, there will be an initiation. Okay. So when we talk about initiation, we are talking about a mutation which is going to happen in the DNA, and that is leading to the formation of cancer. Okay. Once the initiation has been taking place, means the normal cells have been converted into cancerous cells, next comes the promotion and the progression. And the promotion and the progression depends upon different type of regulatory, we can say, factors. And the most important factors, if we look at, is that they resist the cell death. Cell death is what? Apoptosis. Okay, so they are resisting the apoptosis. Means if the damage is taking place or the in the DNA, the cell repair is getting hampered and there will be no apoptosis. Okay. Now, after the this cells has been proliferation, then they give rise to a large number of daughter cells. Okay. Next, the role is played by different types of receptors, and these receptors plays in the role of sustaining the signaling pathway. Okay, when they are evading this different types of growth suppressors, they are also inducing the angiogenesis because they will be needing an oxygen to survive. So every cells need an oxygen to survive, and how they are going to get this this oxygen to survive? They are doing it by angiogenesis. Angiogenesis is the formation of the blood vessels. So when the cell is growing, when the cell tumor formation is taking place, they are inducing the angiogenesis, that is the formation of the blood vessels, so that they can get a supply of all the growth nutrient which is required for the growth. So they, apart from that, because of their this capacity, they are also evading the surrounding tissues. Okay, so there are different level of signaling pathway which controls the growth of the cancerous cells. Okay, and the most important aspect is that there are different types of receptors which gets activated once the cancerous cells you can see proliferation is going on. So if we compare this normal cells and the cancerous cells, microscopically, if you see any cells, they will be having an, you can say morphologically, they will be different from that of your normal cells. Okay, they will be having your large nucleus or prominent nuclei. Sometimes there will be difference in the structure feature also. The cytoskeleton will be abnormal. They will no longer be requiring an encourage dependent growth because they are evading, they have a very high mass, cell mass. So they will not require any adhesive or adhesiveness, means encouragement with the surrounding tissue. They will be grow independently of the density. So if you will do the karyotyping, okay, karyotyping is a technique. So you will see there will be a chromosomal abnormalities can easily be seen in this type of cancer cells. So if we compare normal cells and cancer cells, you can see their abnormality can be seen at the nucleus level, cytoskeleton level, and their growth is, or you can say, independent of the density. Okay. And this is this green, which I mentioned, that basic fibroblast growth factor. These are different types of factors which play a very important role in signaling pathway. I hope you will be having understanding of the cell signaling. So cell signaling is a regulatory pathway. There are different types of cell regulations which goes inside the cells, and each regulations is controlled by a certain factors. And these factors, if they are, you can say, any deviation is there in the factors, then your cell regulation gets hampered. So basics of cell signaling is very important when you are working in the cancer biology, okay? Because it's all about signaling, okay? Signaling, the replication, the transcription, how it is going on, all is based on this only. So you should have a knowledge of the cell signaling pathway, okay? So I was talking about the first step, that is the how can normal cells change into your cancer cells. And the most important aspect is mutation. Okay, so can you define what is mutation? What is mutation? It's a very common terminology you have come across in your biology also. Mutation. Changes, where the changes are right. Okay. Okay, so you have mentioned very clearly that it is an alteration in the genome, or we can say the changes which is happening in our DNA. Okay, so DNA, any change in the DNA is going to cause the mutation. Okay, so you all know the DNA is having a genes. So this alteration, or which is caused by this mutation, leads to, you can say, as I have told you, different growth factor receptors. You know, receptors are mostly the protein moieties. Mostly the protein moieties, these receptors are there. And the proteins are encoded by our gene. So if there is any alteration in our genetic sequence, it is going to affect the protein structure also, and its orientation also. So if there is a mutation, it is going to affect the receptors also. Signal transduction genes are there. There's cell cycle regulatory genes are there. DNA repair genes are there, and there is a apoptosis controlling genes are there. So if there is any changes which is going to happen in this cell cycle regulatory pathway, or DNA repair gene, or the gene controlling apoptosis, this is going to hamper your cell division. Okay, this is going to hamper your cell repair mechanism. So if this type of regulation is getting hampered, so what will happen? The cells will not be able to regulate its growth. So normally it has been seen that mutation is one of the major cause of this type of growth. And how this mutation occurs? Sometimes these mutations are, you can say, genetic. Okay, genetic means they are already present in the germline. And sometimes this mutations are somatic mutations. Okay, so when we are talking about this mutations can be differentiated into two. The first is your germline mutations, where the change in the DNA sequence that can be inherited from any of the either parents. Okay, so this is called inherited type of mutation. And normally this type of cases, the patient inherits one non-functional copy of a tumor suppressor gene, and when the second comes, then only the cancer is induced. Okay. Next is a somatic mutation. Somatic mutations, the mutations which is happening not in the you can say inherited gene, but it is happening in the cells other than that of sperm or egg. Okay. Somatic means other cells not the germline, that is the egg or the sperm which is fusion takes place and the genes are inherited. Not in that, but in the somatic part. Other do mute your mic because it's really a problem. Like in the middle of I get a noise. So till now it is clear what I'm telling. Is it clear? Should I proceed? Okay. So these mutations which I have talked about can be a germline and this can be somatic. So germline, you all know that the person which inherits from their parents, and the somatic mutations, the mutations which are in the cells other than that of sperm and egg. And so these mutations which takes place in the cancer cells, normal cells will not be having this type of mutations. It has been seen that normally the major cause of the cancer, the mutations happen in the somatic regions. Okay, so the somatic mutations normally cause the major types of cancer. Only 10% of human cancers which occur are because of this hereditary basis, that is because of this germline mutations. And rest 10%, 80% is from somatic, 10% is from inherited, and 10% is the cases where both type of mutations have been taken place. That is if the person had inherited both the germline mutation and the somatic mutation. Okay. Now, this is a DNA sequence I have given you in front of you, and these are, if you see, this is your sequence, DNA sequence, and this is a tumor cell DNA. Okay, so if you look, compare this to sequence, healthy cell DNA and the tumor cell DNA. Can you tell me where the mutation has been taken place? The sequence has been given here. Okay, GTA, GTT, GGA, GCT, GGT. So can you tell me at which point the mutation has taken place? Only one answer I have got till now. Compare the sequence, okay? Compare the sequence and do reply in 3, okay? Don't write T. You know the sequence, how it is read. Amino acids are coded how? Don't write T. Write the sequence. The sequence which is coding the amino acid, okay?
It's ma'am GTT.
Right. Okay. So as you have all mentioned, so earlier in the healthy cell, the GGT was coded. Okay. So always whenever you are replying, do reply in the sequence which is coding an amino acid, because if you're writing a T, none of people will be able to recognize that where the T is lying. So write in the coded form. Okay. That's it. So here, most of you have mentioned very rightly that GGT was the healthy cell DNA, and the sequence where the mutation takes place, it was GTT. So imagine the healthy amino acid, the healthy cell DNA was coding for glycine, and just one mutation has resulted in the coding of the amino acid which is valine. Okay, so you can imagine if such type of mutation is happening, so what will happen? The protein which is going to be encoded, it can lose its functionality. Okay, is it clear to what I'm trying to tell? Because for protein to be functional, its sequence are very important. Okay, so if there are any changes in its amino acid sequence, its functionality, its biofunctionality is going to change. Okay, it can happen that it can be downregulated, means it is not be you can say not too much effective, or sometimes it can happen that because of this, overregulation can also take place, overexpression can also take place. Okay, now, so when the mutation is happening, there are certain lot of research has been done in this area, okay, to identify the genes. Okay, and it has been found that there are certain cancer critical genes. The name has been given, the cancer critical genes. And what are these cancer critical genes? These cancer critical genes are the class of genes which has been showing a lot of, you can say, A lot of cancer cell studies have been done, and they have defined that these are the class of gene which are related with the onset of the cancer. Okay, and these, sorry, and if we divide them, they are basically, majorly there are two types. Okay, basically there are two types. Another, the caretaker gene, or we can say DNA repair gene, is also one of the class of a gene. But the majorly, the cancer critical genes are of two types, and they are known as tumor suppressor gene, or you can say proto-oncogenes. Okay, so these are two major type of cancer critical genes. Okay, so if we talk about the proto-oncogenes, their normal function is that they promote cell survival or proliferation. Normally, in the normal cells, these will be related with the promotion of the cell survival or proliferation. And tumor suppressor gene, as you know, the name itself is that they inhibit the cell survival or proliferation. Okay, so if there is any changes in this two type of gene, that is in the case of when a proto-oncogene is converted into an oncogene, or a tumor suppressor gene is being, you can say, is expressed or not expressed fully, it will result into your onset of a cancer. Okay, so normally it has been seen that this tumor suppressor gene, because it is playing a role in the, you can say, proliferation, inhibiting the proliferation. So it plays a very important role. You know, the cell cycle, we have G1, R, S, and G2 phase. Okay, so every cell cycle, every cells have a particular cell cycle. So there will be G1 phase, there will be R phase, replication phase, there will be S phase, and a G2 phase. You all have a knowledge about the cell cycle. You know cell cycle?
Yes, ma'am.
Okay, so normally it has been seen that because the tumor suppressor gene has a important inhibition, so it inhibits in the case of your at the replication phase. Okay, and the proto-oncogenes or the oncogenes, one of the most important oncogene is RAS protein. It affects the G1 phase. Okay, so it has been found that these are two type of you can say phases of the cell cycle which gets regulated during the oncogenes and tumor suppressor gene. If we talk about the caretaker gene, okay, the caretaker genes are normally the repair or the preventing DNA damage genes. Okay, so what happens if the mutation is taking place in this type of genes where the factor is going to regulate the DNA repairing mechanism? It will get hampered. Okay, so this type of caretaker gene mutation normally takes place by a point mutation, or you can say a methylation, or you can see a deletion. And in the case of this tumor suppressor gene, it has been seen that the mutation which is taking place in the case of tumor suppressor gene, it is also by deletion, point mutation, or methylation. But the mutation which takes place in the oncogene is totally different. In this type of oncogene, the mutation is by chromosomal translocation. You know, in chromosomal translocation takes place, and also from amplifications, and sometimes point mutations have also been seen. Okay, so if we compare this to, that is your tumor suppressor gene and the oncogene, because there are two major cancer critical genes, we will find that this oncogene type of mutation, or you can say gene, is a dominant type of mutation. Means if it is happening in the normal cell, it will directly lead into a cell proliferation. Means it is an overactivity mutation. It is a mutations which are genetically very dominant. So if the mutation is taking place in the proto-oncogene and is converted into oncogene, it will be directly relating to your abnormal cell proliferation. But when your tumor suppressor type of critical gene is involved, it is not that dominant. It is a recessive type of mutation. Okay, mutations are genetically recessive. Okay, so what happens? It is normally termed as underactivity mutation. Okay, so it is termed as underactivity mutations. And here what happens? there will be a first mutations. So one mutation will be taking place. So in the one copy of a gene, so there will be no much effect. So cell will not be entering into a cell proliferation stage. Okay. If the second mutation is taking place, means if the second mutation is happening in the second gene copy, then only the, you can say, the tumor suppressant gene will be ineffective. Okay, so once the tumor suppressor gene is becoming ineffective, what will happen? The cell will not be undergoing your apoptosis and it will be stimulating the cell proliferation. So you can imagine, if you remember what here I have shown, the replication part was blocked because of the tumor suppressor gene. But what will happen if tumor suppressor gene is getting mutated after two mutations? It is not a dominant type, it is a recessive type. So there will be, when the mutation is happening in both the copies of chromosome, then only tumor suppressor gene will become ineffective. So what will happen? Uncontrolled replication will be happening and uncontrolled growth will be happening. Okay. And in the case of your proto-oncogenes, it is a dominant, means a single copy is having a mutation, it will resulting into your cell proliferation. Okay. So if I give you an oncogene type of examples, if you find that there is a chromosomal translocation which has been seen in the case of chronic blood cancer, that is of ABL and BCR. Point mutation is in the case of RAS gene, which is involved in the case of colon cancer. Deletion is the ERB gene, which is faced in the breast cancer. Then amplification in the neuroblastoma, insertion and activation in the case of acute blood cancer. So these are different type of genetic changes which has been seen in the case of oncogene type of generation. Now, next example I will be giving you about a tumor suppressor gene, that is a P53. P53 is you can say tumor suppressor gene, and its activity is that it stops the growth of a tumor. Okay, so normally it has been seen that P53 abnormality means if the P53 is getting a gene is including the mutation, it is the cause of 60% of the most of the cancer. And P53 is a nuclear transcription factor with a pro-apoptotic function, means it is related to the apoptosis, means the cell death. Okay. So in the normal condition, what happens? It will normal condition when the normal cell is going on, if P53 is expressed, so it will be causing a cell death. It will be causing angiogenesis, differentiation, DNA metabolism, give it a different role. Okay, different role in a different cell signaling pathway. So how it does look like? This is a sequence of P53. So we have a transactivation domain here. This is a proline rich, green one is proline rich gene. This is a DNA binding domain. This is a nuclear localization signal. Then we have different export signal. NES is a nuclear export signal, and this is a basic domain. Normally, this is a normal sequence for the P53. It has been observed that 94.5% of the mutation, which is the cause for the 60% of the cancer, happens in this DNA binding domain. Okay, so the core domain where the DNA binds is the DNA where the DNA binding domain is there. This is the cause for most of the 60% of the cancers, because 95% mutation occurs here. Okay. So in the case of lung cancer, if you see their mutation has been seen in the area of 157, 158. These are the sequence, the reasons where the mutation has been taking place. And reasons for this gene alteration are missense mutation, frame shift mutations. So there are, most of the time, the reason of this DNA binding domain is responsible for this type of your mutations. So normally it has been seen that recent report by International Agency for Research on Cancer, they have found that this P53 contain about 17,689 somatic mutations. Imagine the level of somatic mutation which is happening in the P53, and 225 is the germline mutation which has been reported till now. So 97% of the p53 mutations are in the core binding domain. 75% of these mutations, out of this 97%, 75% are because of the missense type of mutation. Okay. So normally, how this p53 works? P53 works in is located in the chromosome 17. Okay, and it binds to the DNA. In turn, stimulates another gene that is p21. So it binds to DNA and you can say regulate the production of a protein that is p21, and this p21 reacts with CDK, that is your cell division stimulating protein, and then because of this the cell division is stopped, so cell cycle arrest takes place because of this p21. So what happens when there is a DNA damage of the p53, wild type p53 is entering into a mutant phase? So what will happen? So this p53 will not be able to bind to DNA because the mutation is taking place in the DNA binding domain. So if the mutation is happening, so this p53 will not be able to bind your p21, and if they are not binding to the p21, they so if they are not binding to the p21, so what will happen? Then they will not be able to help in the cell cycle arrest. Okay, this is one approach. Okay, where we are arresting the cell cycle. Another way is that they are entering into the apoptosis. So once they are mutant, becoming the mutant, they are this apoptotic pathway, cell signaling pathway is also getting stopped, and they are entering into the oncogenesis. So imagine if there is mutations like this, which are you can say leading to your stopping your cell cycle as well as apoptosis. So they are regulating the pathway of the you can say cancer cell towards the progression. Okay, so these type of mutations will help in the progression of the cancer. Okay, so they are not directly involved in the cancer cell formation. They are involved indirectly by enhancing the progression. Okay, but when we are talking about the proto-oncogenes, they are directly involved because the normal cell is getting converted into your cancerous cell. Okay, so they are directly involved, and here they are helping in the progression of your cancerous cells. So so parameters which need to be examined when you are working with this you can say cancer cells is that obviously the cell growth, you have to see about how the cell progression growth is taking place, how the transformation of the normal cell to the cancer cell is taking place. So initial examination, cancer biology deals with all these parameters, that is how the cellular growth is taking place, how the invasion is taking place in the surrounding tissue, how the angiogenesis is taking place. Next is that what is the difference between the normal cell and the cancerous cell? Here you will be working on the genetic level, means you will be going on the genetic sequence, how the genetic sequence of the cancerous cell is totally different from the normal cells. Okay, once you have this genetic sequence, then you will be progressing towards the cell signaling, how the cell signaling is different from a normal cells and how the cell signaling is different from the cancerous cells. Okay. Another aspect, which is the major factor why the cancer cell treatment is becoming very tedious, or you can say the cure is not that visible, is because of the metastasis. Metastasis is the term which is related with the spread of the cancer cells. Okay, so whenever there is a progression in the different you can say organs in the body, then it is called a metastatic stage, and because of the metastasis, the cure of cancer is becoming very difficult, because the metastatic phase of different type of cancer cell is totally different, because the you can say the factors which are involved in different type of metastatic cancer cell is varies from person to person. Okay, so how this done, this cancer cell staging is done. This is done by TNM method. The TNM is T stands for the extent of the primary tumor, and stand for the if it has been inverted into the lymph nodes or not, and the third is the presence or absence of the metastatic stage. Okay, so depending on that, the stages are defined: stage 0, stage 1, and the stage 4, you all know it is a very advanced disease where the cancerous cells have invaded your different organs. Okay, and it is the stage where the treatment is, you can say, very limited, and the cure is very limited. So with this, we will now be talking about the scope and opportunities. Hope till now it was clear to all of you about the basics of the cancer cell. It was clear to all of you?
Yes, ma'am.
Yes, ma'am.
Okay, thank you for your reply. So let us talk about the scope and opportunities. So before going into the scope and opportunities, I would like to tell you that cancer biology is a pure research-based area where a lot of research is required. Okay, and it requires a lot of technical skills also. Okay, skills means the lab skills as well as the different instruments which are required for detecting the pattern of growth, to check the different proteins, isolation. All these are very much required in this area of cancer biology. So normally this is related to the R&D section. Okay, so every industry which are working in this area of cure of the medicine for the cancer, they have this R&D section. So if you like to work in the cancer biology research, you have an opportunity in the R&D sections. Government organization, if you talk about, there are a lot of cancer institute. There is a Tata Research Institute, Cancer Research Institute, and every research institution is nowadays having this lab on cancer. Okay, and they are working on different cell lines. You know, we will be having a knowledge of the cell line, animal cell culture. So they are using the cell cultures to check, or you can say to study the different type of medicines on how these cancers can be cured based on and also they are understanding that how this medicine are working at the cellular level, molecular level. Okay, so this is one of the way that you can join this government organization. There are 3 ways you can join, because most of you will be from BSc, MSc, or MTech, BTech. So you can join this government organization as a GRF, that is a Junior Research Fellowship, or a Senior Research Fellowship, or if you are a PhD, you can join them as a scientist. Okay, but for BTech, MTech, BSc, MSc, scientist position will not be there. Okay, you will be joining as a project fellow, or you can join as a PhD program of these government organizations. Okay, for that, if you want an independent fellowship, then you can qualify the CSIR, GATE, DBT, ICMR, different types of fellowships are there. And if you are unable to qualify these exams because of any certain reasons, you can join through a project. So you can check different types of opportunity on this different institutes. They will be having a projects, and these projects are funded by different types of organizations like DST, DBT, Spark, different type of organization fund this type of project, and there you will be getting a scholarship or the fellowship, and through that you can work in this government organizations, right? Now, if you're working in the R&D sections of the companies like the biotechnological or pharmaceutical companies, mostly it will be really difficult that you will be entering in the R&D section directly. For that, they will be needing, just after BTech and MTech, you will not be able to get this R&D sections. Okay, here you will be requiring a skills and expertise, and for that, it is preferable that you get a hand-on experience, because you will not be much exposed during your course, during your 6 months training or 1 year training, will not be much exposed to that. Okay, so to get this training or different type of internship, get in hand-on experience, because when you work with the cell lines, it required a lot of expertise. Okay, so it is always required that you have expertise, then only you can enter this biotechnological companies and pharmaceuticals companies which are working in the R&D section. Okay. And if you are PhD, you can get the job in these biotechnological companies. If you are postdoc, that is most preferable, because more and more knowledge in this area is going to help you and get you more career option in comparison at BTech and MTech level. Okay, clear what I'm trying to tell? Is it clear to what I'm telling? That in this area, in the cancer biology, if you want to work, you should have a proper skill and expertise, because here almost most of your time will be in the lab only. Okay, so if you're not having a skill, if you don't know how to subculture a cell line, if you don't know how to extract a DNA, if you don't know how to analyze a protein, if you don't know how to do a cell counting, if you don't know how to perform this different types of instrumentation, it is really difficult that you will be getting an opportunity. So try to get the fellowships, do PhD or do internship, or do training in this area. Different institute nowadays, they are giving the training, giving out training opportunities in handling this cell lines and performing different types of tests like mass spectroscopy, protein analysis, and all this. So try to get it. Okay, try to join this internship trainings, and then only you will be getting a more job prospects and the career opportunities. Okay. Apart from that, if I talk about, there are a lot of academic roles are also there. So you can join as a tutor or instructor on the online platform if you're having very good knowledge about the cancer biology. If you're MTech from a private MTech, then you can join these private colleges and the online platform. They will be preferring you. PhDs will be getting better options from the MTechs in the universities, colleges, and the NITs. And postdoctorate is very much necessary if you want to join this IIT, ISR. So postdoctorate means you have a post PhD experience postdoctorate is post PhD experience is very much required if you want to enter into the academic area. Okay, or you can join as a project as a scientist in this government organization also. Then also they will prefer PhD and postdoc particularly in the case of cancer biology. Okay, so cancer biology is more of advanced stage where you'll be getting a better job prospects. Okay. Okay, thank you everyone. This was all about today's lecture, and I hope I tried my level best so that I can give you a short overview of what is a cancer biology and how you can have options if you want to work in this area. Okay, and I request you all to give you can say a review on this link which I am sharing with everyone. So because we will love to hear your experience, and do give your reviews. If you have any question and query, you can ask me. Okay.
Hello, ma'am. Hello, ma'am.
Ma'am,
Yes.
ma'am, can you please tell that I am currently, I have completed my BSc in Medical Laboratory Sciences and I wanted to pursue my cancer biology.
So
So
you are
can you please? Yes,
BSc in
ma'am.
what? BSc in?
Medical laboratory sciences.
The voice is not clear. Can you write in
Um,
the chat box?
yes, ma'am, sure.
Hello, ma'am.
Yes.
I have question regarding caretaker gene. So, how caretaker genes regulate methylation of DNA?
Caretaker gene. Sorry.
Ma'am, you have given the idea that a caretaker gene involved in the methylation of DNA.
Okay.
So, can you elaborate?
So you are talking about a DNA repair mechanism, right?
Ma'am, you have explained three: tumor suppressor,
No,
oncogenes,
I
and
am telling
caretaker
about
genes.
it, how this mutation is happening. Okay, like mutation is happening in the three main cancer critical genes are of two, so tumor suppressor, and one is your this protoncogenes. Apart from that, you know, there is a DNA repair mechanism which goes on inside the body. Okay, so what is the DNA repair mechanism? Is that if there is certain mutation is happening place, this DNA repair mechanism, so DNA repair signaling pathway, it is the checking is going on. Like DNA, the body itself checks that mutation is happening and it's repair it. Okay, it will repair it. So what happens if the genes which are involved in the DNA repair signaling pathway? Okay, so if a gene is you can say responsible in a DNA repair pathway, what will happen if that gene is getting you can say mutated? How this mutation is taking place? Mutation can be a point mutation, as I have mentioned you, and sometimes the methylation of DNA occurs. Okay, so when the DNA methylation occurs, DNA will not be able to encode the genes, perfect genes. So what will happen? The DNA repair mechanism will getting hampered, and the mutation which has taken place already will be start going on. So there will be unregulated growth. Are you getting my point? What I'm trying
Yes,
to
ma'am.
say?
Yes, ma'am.
Ma'am. Hello, ma'am.
Yes, someone has mentioned about that. Yeah, can you please suggest? If you
Ma'am.
are an MSc, what I will try, you try to get entered through the biotechnology, okay? Because in the biotechnology, only when you're doing MSc biotechnology, then you will be getting an opportunity. Like you can apply for the GRF, SRF positions. You can qualify CSIR also, or you can go for GATE also, and you can apply for ICMR GRF also. You can go for DBT also. So there will be a lot of option if you are going through this MSc biotechnology, and from there, from third semester, okay, when you will be in the third semester, start preparing for this competitive exams. By the fourth semester, you will be able to give you GATE also and all these 4 exams. And if you clear it, then you can enter any of the cancer research institutes. There are a lot of cancer institutes are there which are working in this area, particular area. Apart from that, you can, if you're not qualifying this exams also, because of any reason, you can directly join through a project. Okay, so these institutes have the projects. They give an advertisement, like they have a GRF position, they have a SRF position. So through that you can join it. Okay.
Ma'am, I'm currently applying for cancer biology in UK. So that's why I was a bit confusing that, and I'm also having seat in clinical research in DY Patil University.
Actually, your voice is not clear. Can you write it?
Yes, ma'am.
Excuse me, ma'am.
Yes.
Ma'am, I'm currently pursuing my degree, BSc second year in genetics and biotechnology.
Okay.
For choosing cancer biology in my master's, what are the entrance exams should I write, ma'am?
Pure microbiology.
I'm from genetics and biotechnology, ma'am.
And so you are doing a BSc right now. right?
Yes, ma'am. Second year, ma'am.
Yeah, you can go after in the third year, you can apply for this. There is an entrance exam, MSc Biotechnology, which is a combined exam by DBT GNU. Okay, so you can give that exam also. There they have around 30 institutions, and they give an option for this MSc Biotechnology, and they give you fellowship also for that. So you can go for that also. And apart from that, you can go for ISC. Even ISC also offers your different types of MSc joint entrance, MSc PhD. And in the IITs also, they have a MSc PhD. So you can join that also, okay, after your BSc.
Okay, ma'am. Thank you, ma'am.
See, most important part is that you have to be very clear about the basics, because everywhere you are going to face this you can say objective questions. There will be a written followed by an interview everywhere. So you have to clear your basics. Unless and until you are not clear about the basics, you will not be able to answer those written exams, because after qualifying also, most of the IIT, ISC, JM, CSR, or your IISER, all will be having their own written exams and your interviews. So try to clear your basics as much as you can. Try to clear your application part also, because they will be mostly asking for the application oriented questions. So try to
Hello, ma'am.
have a clear vision about that.
Hello.
Yes.
Hello, ma'am. I am right now MTech biotechnology student, and I want to do some internship or hands-on training related to or relevant to cancer biology. So, can you suggest me some institutes or laboratories regarding all these things?
Yeah, you can try for different institutes like it. You can Tata Memorial is there. They are also offering certain internships. Then you have JNCSR also. You have CDFD, that Central Drug Institute, which is in Lucknow also. CDRI is there. So there also you can go for internship. Then CCMB is also offering. They have their internships. Or you can do one thing. You can write to different professors or the scientists which are working in this area, and if they will be having this internship or they will be having this, so they can call you for that, for the internship or training also. But you have to look out that which professor or the scientist is working in this area. Prepare your one letter, good letter, cover letter, and then send to them. And if they will be having, they will be calling you. Okay, so you can approach this way also. Like prepare a cover letter, write about your interest, and then convey this to different scientists and professors. If they will be having this opportunity in their lab, they will be giving you 100%. Okay. So you can contact CCMB professor, CCMB scientist, CDRI scientist. You can contact the professors, okay, or the scientists.
Hello, ma'am.
Yes.
Ma'am, right now I'm pursuing my MSc microbiology. After completing my microbiology, ma'am, I done cancer biology, a handful training, some institute. Can you suggest some institute name?
You are MSc microbiology. Do you have gone through this cancer biology? Is your subject or any lab exposure is there for your cancer biology?
No, ma'am.
Okay. So for microbiology students, I think it will be a bit because cancer biology is not a part of your curriculum. Okay. So normally it will be a bit difficult for you because you will not be exposed to the lab experience of how you can handle the cell line and how you can proceed. So better you can join certain certification course related to that, so that your basics will be get cleared about what is it, how cancer biology. Because whenever you are going to approach someone for internship or a training, they will be looking after you that you have that basic knowledge. Are you getting my point? If you come to me like give me internship, so what I will be asking about the basics of that cancer biology. So those who are not having this basics of cancer biology, first of all try to get clear about the cancer biology basics, then only you approach, then it will be preferable, because if they are going to take your interview, will not be able to handle those questions. Okay. And if a person which will be having knowledge of cancer biology, most probably they will be preferring that person in comparison to you. Okay. Even in the internships and the training also. Okay.
Okay, ma'am. Thank you.
Any more questions? Okay, do give your review on this link which I have given you. Someone has mentioned that his MSc in applied cancer and research in. See, if you're getting funding there, you're getting a fellowship, and the lab in which you are going to work with the lab in which you are getting this opportunity, okay, because you will be applied in some lab only in the foreign, in the UK. So if the publication of that lab is good, publication, you don't know what I'm trying to tell. The paper, research paper, the publication of that lab is good. The lab is having all the equipments related to the cancer. The funding is good, means you're getting a better fellowship if you're joining there. I will be saying that will be much preferable in comparison to if you want to pursue in India, because in comparison to India, the cancer research